mitosis, not meiosis like it is for us. We undergo mitosis and we get anywhere from 2 to 6 eggs. Right? That was our female side. Then on our male side over here, we have on the scales of their cones microsporangia, because they're making microspores. So inside here, these little dots right here are going to represent our microspores, which then develop into a male gametophyte, which in the case of seed plants is the pollen grain. So that's what you see here, these pollen grains. They develop from those microspores. And in this case, all of the male gametophytes develop into pollen grains. We don't have a situation where some of the megaspores over here disintegrated and only one developed. All of the microspores develop, and they're all going to become pollen grains. And then what happens after that? Typically speaking, in our gymnosperms, the pollen is going to be windborne, so it gets transferred from the male parts over to the female parts, and it usually gets caught by some sort of sticky drop that's on the female cone. And then once the pollen gets there, then it starts to form the tube. So remember, that's one of the shared drive traits for the seed plants, is to have pollen, but also form that pollen tube, because now we're getting into some of the plants that don't have swimming sperm. They have non-motile sperm, so that's got to be delivered somehow. So the tube is going to go down to where the egg is located, and it's going to release those sperm cells right where the egg is, so that the egg can get fertilized. Right? Now in the case of these gymnosperms, one sperm will actually do the fertilizing. The other one, if it doesn't get there first, it doesn't get to do anything. Right? But in this case, one of these will fuse with our egg. When it does fuse with the egg, it becomes the zygote, right? Which will then divide and grow and become the embryo, which is inside of the seed. Right? So remember, there's 3 parts to a seed. There's the embryo, which is the baby plant. You have nutritive tissue that's constantly supplying food to the embryo so it can survive until it's able to germinate and make its own leaves to do its own photosynthesis and its own roots to get its own nutrients and water. And then you have a seed coat on the outside of that. Okay, so if we look at our gymnosperm seed, because we're going to do comparison here today between the gymnosperm seed and the angiosperm seed, there are some similarities and there's some differences. Okay? Whether you're talking about a gymnosperm or an angiosperm, you're always going to have an embryo, a seed coat, and some sort of nutritive tissue. Okay? What's going to be different is what the nutritive tissue is. Okay? So for the case of gymnosperm seeds, your nutritive tissue is going to actually be the female gametophyte. That is the source of food for that developing, growing baby plant. Okay? When we get into angiosperms, and we start talking about this process of double fertilization, this other sperm, this sperm is going to fuse with another cell, and that substance is going to become the nutritive tissue. It's called endosperm. So we'll get into that one later. So there's a difference in the nutritive tissue. They both have nutritive tissue, but the embryo needs that. But what that nutritive tissue differs. For gymnosperm seeds, it's the female gametophyte, and for the angiosperms, it's going to be something called the endosperm. Okay? So this is kind of how the process goes here. We've got our scale for a female cone. This is going to be our megasporangia. There's the megaspore. Okay? That is kind of, if you take this picture and you turn it upside down, that's what this is right here. You can see the pollen grains have landed here on the sticky drop here. The pollen grain is then going to go down into this hole right here. We won't get into what that's called. Goes right into the hole right here. These, you can see, are now starting to degenerate. Those are the 3 megaspores that are going to degenerate and disintegrate. This is the only one that's actually going to develop. That megaspore will become the female gametophyte, which is now this. Right? So we've gone from this small thing right here to this big thing taking up pretty much all the space. Right? And it's already divided to produce an egg. So there's our egg cell right there. There's our pollen grains, and they're now putting their tubes out to come down here to fertilize the egg. This is where fertilization occurs, where the sperm gets delivered and it actually fuses with the egg. That then forms the zygote. Okay? And that zygote will become our embryo. Okay? And we talked about the different parts of the embryo as well. Okay? The other parts here, you can see the female gametophyte still here. That's our nutritive tissue. Okay? And the seed coat actually forms from something completely separate, this thing right here, which was around the megasporangia. It's something called the integument. Okay? So that makes your seed coat. So just bear in mind, I always want students to know that when the egg gets fertilized by the sperm, that cell that forms is not the seed. It's only part of the seed. That is just going to be the embryo of the seed. There's other things that actually form the seed in addition to just the sperm and egg. All right. Now, within the conifers, there is a clade here that is referred to as the knee-tails. This has recently been rearranged. This used to be its own little grouping, and they're still having a lot of trouble figuring out where this group is supposed to go because it has a lot of similarities with flowering plants. Okay? It doesn't have flowers, but it's gonna have double fertilization, it's gonna have vessel elements, all things that all the other gymnosperms don't have. But based on structure, molecular data, analysis, things like that, keeping in mind there's debate here. There's differences of opinions in how these should be organized, but to date, right now, they have grouped it together within the conifers based on molecular data. Right? So we're considering it a clade within our conifers. Okay? Now, if we look at that group, I've got kind of highlighted a feature in particular that defines them, and this is going to be their shared derived trait. Out of all the gymnosperms, this is the only group that actually has vessel elements. Those are the more specialized water conducting cells that we talked about when we discussed the xylem. You had tracheids, you had vessel elements. Do these also have tracheids? Yes, because all vascular plants have tracheids. It's just that some of them also have vessel elements in addition to that. And none of the other gymnosperms do, but this one does. The only other group that has vessel elements in it are angiosperms, and not even all of those have it. So it's kind of unusual. And the other thing about them is not just the fact that it has vessel elements, because that in itself is not a shared derived trait since angiosperms have it as well. It's what they look like, right? So remember, this is something that's moving water from one cell to another via basically a tube. I don't know if I trust this marker. Let me check real quick to make sure this board has been having some issues. All right, we're gonna go with that one. Okay. So we've got one cell here. We've got another cell on top of that one. Connector down here. Okay, between this cell to this next cell, you've got to have openings here that run at the end walls of the cells to allow the water to go through. Okay? If you look at these, they have pores, and so it looks a little something like this. We call it porous plates. Okay? We'll do the same thing down here. If you look at the ones in the angiosperms, they're a little bit more defined, a little bit bigger. So I'll draw this one here. They may have what are considered more bar-like openings that look sort of like this. Or you may have a situation like this where it's just one big opening that connects each one of the cells to each other. They don't have these little pores. Right, the same way that this is. That's unique to the knee tails. So if they've got vessel elements, take a look and see what the connecting points look like between the cells to see if it is something like this where it's going to belong to the knee tails. They have simple leaves, although what those leaves look like is going to vary tremendously across the group. Some of them have leaves that look like little scales. Some of them have normal looking leaves, like you see the medium up there. And then we're going to talk about one called Lilichium that has only 2 leaves in its entire life that just keep getting bigger and bigger and bigger and bigger and bigger. Right? The other thing they have is they have a form of double fertilization where one sperm is going to fuse with the egg. The other sperm that's delivered by that pollen tube will fuse with a different cell. It's going to fuse with something called the ventral canal nucleus. But nothing really happens beyond that. It may divide mitotically a few times, but it doesn't really develop into anything that the seed is going to use. Okay? Whereas in the case of the angiosperms, it's going to develop into that nutritive tissue. So even though they have this similarity, based on that difference alone and some of the other differences that they see in the process involved, they are believing now that these 2 processes were independently evolved separately in the 2 lineages. We have a form of double fertilization in the knee tails, a different form that we're going to consider a shared derived trait for the angiosperms. And we'll go into that one in much more detail than we're going to do with this one. But I do want you to at least know that they have one, just so you can understand how close they seem as far as sister clades to the angiosperms and why they're so difficult to figure out where they're supposed to be placed. Now, chances are you're not going to see anything in this group around here in Tennessee because they're very highly specialized. There's only 3 families, and within each one of those families, there's only 1 genus each. And so we're actually just going to look at the genera and use those as examples for the groups. There's only 80 living species. The 3 lineages, or the 3 genera that exist is Needham, Ephedra, and yes, that is the source of the medication, and something called Welwitschia. And they're very highly specialized for their respective environments. There are some that are tropical vines, right, live only in tropical regions, and you can see those in the red areas, or excuse me, down here, and the blue areas. You have some that are desert dwelling shrubs, and that's the only place they're found, like the red regions right here. And then you have this one right here, Welwitschia, that's only found in a single desert. And that's it, one place, and it's right there. That's the only place that one occurs. And it's a really weird and unusual looking plant, so we'll look at that one in a little more detail. Generally speaking, all of these are really weird looking plants. Okay, so here's Welwitschia, the last one. Okay? And this one only has a single species in the genus. So you've got just one family that's got one genus that's got one species in it. That's it. It's kind of like ginkgos for that reason. So why does it look so weird? Well, this one's sometimes considered, the description is considered octopus-like, because during its lifetime it only ever has 2 leaves, and they're strap-shaped, so they look kind of like this. They will continue to get longer and longer and longer and longer and longer during its entire life, which can be for a very long time. So carbon dating tells us that on average, they live to be about 500 to 600 years old, right? Although some of the large ones are thought to be almost 2,000 years old. They're really, really old plants. And so you can imagine if this just keeps growing and keep getting bigger, it's gonna start to curl on itself, it's gonna wrap around, and eventually it's gonna start to fray and tear and rip. And so it ends up looking like this kind of frayed one you see right here, kind of like octopus tentacles. But that's actually only 2 leaves, what you're seeing right there. It lives in a really dry desert, and so it uses every means that it can to try and get the source of water that it needs. It can catch moisture from sea fogs. It also has really long tap roots, right, that go really deep into the soil, that they can search for any underground water, making it well adapted for its harsh environment. But kind of a cool looking plant, but one of our details that does that double fertilization and has those floral specials. All right, now this is where your test ends. Right, so you can put a line in your notes for this is where your test is going to end. Now what we're going to get into is the next group that we're going to spend the whole rest of the semester talking about. And that's going to be our angiosperms, because about 95% of all plants, modern day species that we see today, are some sort of an angiosperm, some sort of a flowering plant. So this one's actually changed names as well. If you go back into your old Campbell book that you had for Biology 130, 140, they're probably still referring to this group as Anthophyta, but that's actually not the case anymore. That's not the phylum or division name anymore. This one has now been renamed into a group that's called Magnoliophyta. Right? We usually refer to them as angiosperms just for short. It's a really important group, and making this transition into something that's a flowering plant, this was not a one-step process either. You can see all of these different shared derived traits that are associated with the angiosperms that are unique to them. All these happened along this evolution towards this particular clade. So we've got a lot of shared derived traits to go through for this one. But if you know things about angiosperms, if you can think about flowering plants, some of these should be obvious. If it's a part of a flower, it's going to be a shared derived trait for the angiosperms because they're the only ones that have flowers. So we're talking stamens, we're talking carpels. You can't get a fruit without a carpel, so you got to have fruits. So all of that's going to be related to our shared derived trait for angiosperms. So we'll list them first, just like we usually do, and then we'll go through them individually. So first and foremost, most important one is we now have the flower. This is the only group that makes flowers of the 4 major groups of plants. Non-vascular plants, seedless vascular plants, gymnosperms, none of them make flowers. That means parts of the flower like the stamen and the carpel are also going to be shared derived traits for the angiosperms. If you recall in lab, I also talked about how these structures, all of the whorls of the flower are essentially modified leaves. And leaves that in the past have either curled in on themselves and fused together or become more specialized to make these reproductive structures that you see here. In the carpel, The ovary portion of that is usually what becomes the fruit. Doesn't mean other parts can't be involved, but the ovary is gonna be involved in making that fruit. So you can't have a fruit if you don't have an ovary, and you can't have an ovary unless you have a carpel. So that's kind of linked together. They're also gonna have reduced gametophytes. Both the male gametophyte, which is the pollen grain, is gonna be really reduced, and the female one. To the point in the female one that they don't even form archegonia anymore, because they're so small. And the pollen grain is only gonna be 2 cells, and that's it. That's how tiny it is. Very, very reduced. There's some advantages to that. You don't have to expend as much energy to make them, which is always good for the plant. Then we have number 5, double fertilization, which is going to lead us to number 6, which is the formation of endosperm, the nutritive tissue in the seed. And then we have a type of cell that is associated with the phloem. So I told you we'd come back to this one, because we talked in much more detail about tracheids and vessel elements, and we didn't really differentiate too much in the phloem cells between what we call seed cells and seed tube members. But we're gonna have to for this one, because it's different for the phloem cells, right, than it is for the xylem cells. In the xylem cells, if you are gonna be a vascular plant, you're gonna have tracheids. I know if there's a bunch, you have tracheids. You either have only tracheids or you have both. In the case of the phloem cells, you're either going to have seed cells or you're going to have these, seed tube members. So these are actually going to be shared derived traits, and none of the other types of cells that relate to conducting things fell in that category. And so we'll talk about what makes this group so unique. All right, so the most important evolutionary feature for the angiosperms is the fact that they have the flower. And this is thought to evolve in response to selective pressure to transfer pollen from point A to point B. Okay, if you remember, the ancestral means of moving pollen is wind dispersal, which is notoriously ineffective and very risky, because the plant does not have any control over the wind or where it's going to go. There's no guarantee that that will ever get blown to a female reproductive part where the pollen can actually land and fertilize something. And so this is much more adaptive to be able to do this. If you can attract something living to you and specifically have them come to flowers, you know that animal is going to visit another flower. Whether it's the same species or not, that's to be determined, right? But you know it's going to visit another flower. And so you have a higher chance of getting your pollen transferred to another location, to another individual through this process. So when the very first flower evolved, right, in the fossil record, the very first plants that had flowers, we all of a sudden are opening up a whole bunch of new niches, okay, for these organisms to utilize. Because now we have all these potential pollinators that could be involved. Flies and butterflies and bees and beetles and hummingbirds and moths and bats, even other types of mammals like certain types of possums and things like that, that all view and sense the world in different ways, right? So we can become specialists. We can have this one ancestor diversify and branch into all these open niches. And that's exactly what happened. We had an adaptive radiation method in this. So we have multiple pollination mechanisms within different groups of plants. They utilize different pollinators oftentimes. And that led to the evolution of many different chloroforms that are going to be fairly specific to those pollinators. How are they going to sense the world? Where are they going to be attracted to? Now, you don't want it to have smell like rotting weed if you're trying to attract something like a bee that wants a sweet smelling flower, or a hummingbird. You want something that's, if you want to attract flies, then maybe having a carrion smelling or a dead meat smell to your flower would be useful, right? And so this accounts for, in large part, the distinctiveness that we see in a lot of the flowering plant families. If you go into the key in the book, and as soon as you get into the flowering plants, it's going to start talking about flowers. And it's going to start talking about flower anatomy. That's what makes them unique and distinctive. That's how you separate them out into different groups. It's also why your lab exercise, when you had to make your key, and you couldn't use flower terms, you had to use vegetative terms, it's hard to do. Hard to do just vegetative terms. This is something that is definitely used for diagnostic purposes, for classification, and for identification within these angiosperms. Okay, so if you've had animal or plant ecology, you probably have talked a little bit about this, something called pollination syndrome, which talks about how these different colors, these different scents, these different shaped flowers, different sized flowers, are all going to attract different pollinators depending on how they see the world. So for example, here's our carrion flower. As you can guess, it smells like rotting meat. That's the color it looks like as well. It's bringing in flies, like it's going out of style. They really like the smell of it. They think it's something that they can lay their eggs on, and they just come right to the flower and pollinate in the process. This one down here, little brown jug, I actually had to move the leaves out of the way here so you can see they're not colorful at all because they're hidden underneath the leaves. They're not even using visual cues to try and attract the beetle pollinators that come to them, because the beetles are not going to be up here. They're underneath the leaves. They're not going to use visual cues to find this. They're going to use scent cues. And so instead of being colorful, which requires a lot of energy to make, they have a very strong scent. It's kind of spicy or peppery, and the beetles are really attracted to that. The hummingbirds, they see the world fairly similarly to the way we do. Colors look about the same to them as they do to us. They love the color red. Right? Ironically enough, some of these other ones, like for example, bees can't even see that color. So these love red flowers. And you can see they don't have to be real big because hummingbirds don't have to sit on the flower. They can just hover in place. And they've got long beaks that can reach down into really, really deep floral tubes that maybe some of the other types of pollinators can't do. Bees, on the other hand, have land. Right? So we gotta have big enough flower that they've got a little landing pad for them. We've got ones that are white in color because they're active, or they're trying to attract pollinators that are active at night, which you can't see very well. They're not going to use visual cues, they're using scent cues. Most of these things that are pollinated by moths and by bats smell wonderful, really strong and very sweet, right? Because that's their attractant to bring those pollinators in. Okay? And then also in my plant ecology lab, I talk about how the bees are kind of interesting because they can see into a spectrum that we can't. They can see into the UV spectrum, not just visible light like we do. So they're going to see some very different things and some contrast that we may not see when we look at a flower. Now granted, I understand that a dandelion is not a single flower, that's a head. But still, even within the head right here, you can see the bee is actually seeing some additional things, contrast here, different colorations, I'll use quotation marks, on the head than it would be if we just looked at what we see, that yellow coloration. They work kind of like targets, right? It's called a nectar target. It's basically telling you, and I'll show you a picture of this as well. It's basically telling the bee, come this way. This is where your reward is. This is where I want you to be, in the center. And in the process, you're also going to get my pollen. You have no choice in that. If you want the reward, this is where you have to go. And it's kind of guiding it to where it needs to go. So you can see differences in our primrose here. We see yellow, they see this. Okay? This one has only two colors for us. It's got lots of different contrast in the UV spectrum for the bees. And this one, which looks white to us, is actually very vibrant to them, right? If you look at the UV spectrum. So they can, flowers or flowering plants can use this to their advantage, and they can specialize in pollinators to prevent some of the hybridization, right? Because you don't necessarily want to pollinate another species. And if you have your pollinator specific to you, that's not going to be attracted to other types of species, you're less likely to have that problem of hybridization. All right, now this should hopefully be all review from stuff that we talked about in lab, right? And we've been tested on already in our midterm, so my expectation is we know this pretty well. But let's kind of refresh our memory and walk through this. Okay, so in a flower, you have your stalk of your flower down here, that's your pedicel. Then you have the part that the flower actually sits on, which is the receptacle, and then you have a series of 4 whorls, 1, 2, 3, and 4, as you move inward towards the center of the plant. Your non-reproductive whorls, right? If you start at the very bottom down here, the first whorl are your sepals, that if we collectively refer to them all together, we call the calyx, and that's usually how I will refer to it in class as we talk about these families. And they can be green and photosynthetic, they look like kind of leaf-like in some cases, or they can look like petals. They can be kind of petal-like as well. Kind of depends. Some of them even look like little bristles or hairs. If you're looking at, for example, the dandelions have little hairs that were the sepals, so it just depends. So that's our first whorl. The second whorl are the petals. That's the next layer in, and we collectively refer to those as the corolla. Generally speaking, if this is something that's animal pollinated, right, it's going to be fairly colorful, showy, odoriferous. It's going to have rewards, like nectar, for example, that's going to attract those pollinators in. But as we're going to find out, there are still some flowering plants that use the ancestral means of pollination, which is wind dispersal, to do their pollination. And so the ones that are wind pollinated don't usually have these really colorful, showy petals. In many cases, that actually inhibits and makes it harder for pollination to occur because they get in the way. Big petals are in the way of the stamen they're trying to get, or the stigma that they're trying to get the pollen to. And so they have reduced ones, and not a lot of color, not a lot of scent, not a lot of rewards. Those are all expensive to make. And if you don't need to attract wind, because wind doesn't care what the flower looks like, then why make those? Okay? And then if you have whorl number 1 and 2, not differentiated, or at least very similar in appearance, for example, if they're all petaloid looking, very petal-like, then we refer to them as tepals. And we'll see several families that we'll talk about that are going to have tepals as one of their characteristics. Some families, they're differentiated, some families, they're not. Okay, our next whorl in the male parts, what are they called? Stamen. And collectively together, do you remember the collective term for them all together? Androceme, exactly right. Okay. So remember, angiosperms are going to be heterosporous. Just like our gymnosperms, they have separate structures for the male and female parts. They're going to make megaspores, they're going to make microspores. The male part, which are the stamen, collectively called the androceme, produce those microspores. Those microspores then develop into the male gametophytes, which are the pollen grains. And then the male gametophyte makes only sperm. That's why it's male. And then the fourth whorl in the female parts are the gynecium. Collectively, that's all the carpels together. It may only have one carpel, but it could have multiple carpels, and sometimes those will be fused, sometimes not. We'll get to that in a second. But those will produce the megaspores, which develop into female gametophytes, which are going to produce the eggs. All review, right? So far we're good? If not, then at least this is a refresher. Hopefully it's at least a refresher to you. Okay, so that's the flower as a whole. Now let's start getting into some of the structures inside of it, because they're also shared derived traits for flowering plants. First one we'll talk in more detail about is the male part, the stamen. Now remember, the stamen is made up of two parts. You have your filament, which is the stalk that this sits on right here, and then you have your anther on top, which is probably the most important part, because that's what makes the pollen. Okay? And it's going to have these two pollen sacs on it. We won't get into what they're called. They're called theca. Don't worry about that. We've got two sacs. All right, so you can see one right here on this side. We've got another one right here on this side. We're going to kind of cut into that and look down on it, dissect it, see what's on the inside there. Okay, so this is what it looks like in cross section. If you're looking down, you're going to cut it this way. So we've taken the top off of this like this. Now we're looking down on it. That's what this is right here. You'll see, even though it has two pollen sacs, right, what we call two theca, there are a total of 4 microsporangia though. There's one here, there's one here, there's one here, and there's one here. Right? Total 4 in the anther. Now as they mature and they get older, eventually these two will actually open up and kind of converge with each other and fuse together. Same thing will happen with this one right here, so you end up with these basically one chamber here, one chamber here, but they started off as separate microsporangia. Okay? Inside of that, okay, you'll see the pollen grains. So these little dots right here, those are the pollen, and eventually when those are mature, they'll have to get released from the anther, and how that happens will depend. There are many different ways that the pollen sacs can hiss. And yes, that's the same word that I used in lab when I talked about the fruits that popped open, dehiscent. Right? Same thing applies here for the anthers. They're going to pop open, and they're going to dehiss. They're going to send those pollen grains out. They can have slits in them like this, and you can see how that happened over here. This kind of opened up like a flap. This is a slit that runs the whole direction of the anther. Those slits opened up and released pollen. that can have pores in the top or the bottom, or they can have these little valves or flats. We're gonna come back to that one, because there's one family that's one of their distinctive characteristics is that they have these valvular anthers, and that's how the pollen is gonna be released through those theca. Now for the carpel recall is made up of 3 different parts. You have your ovary, that's the portion down here that houses the ovules, which are the immature seeds. You have your style, which is the stalk on top, and the stigma, very on the top of it right there, that has the sticky substances on it to catch the pollen. As you can see, carpels are, just like any other whorl flower, are modified leaves. They used to be leaves in the past, and so we talked about how you have your fern species, your seedless vascular plants. The ferns, the reproductive parts, the sporangia are on the underside of the leaf, oftentimes on the edge of the leaf. If you have something like that here, where we have our reproductive structures on the edges of the leaf on the bottom side, and this were to start to come together, and eventually come together to the point that it fuses, that's what the carpel is. Basically that rolled up leaf. And now your reproductive structures are on the inside of it, in a vessel, right? Which is exactly what angiosperm means. Okay? So at maturity, the carpel body completely encloses those ovules. And so whereas gymnosperm meant naked seed, the sperm part was the seed part, gymno is naked. Angiosperm means seed in a vessel. Okay, so angio is vessel, and sperm is referring to the seed. So that's inside of that. Okay. Now, flowers may have 1 to many carpels. We've kind of started to see that in lab. We've been doing some dissecting of our flowers. One of the things I had you oftentimes do was do a cross-section of the ovary to see if you could figure out how many carpels were there. Is it 2, is it 3, is it 5, is it 10? It depends on the species. But if there is more than 1 carpel, those carpels may either be separate from each other, like they are in the buttercup that I taught you last week in lab. Right, so for example, we've got 3 separate carpels here, 3 distinctive entities right here that are going to be also called pistils, because the pistils are the individual pieces that you see. But each one of those is a simple pistil because it is only made up of 1 carpel. Right? Or you can have multiple carpels that are fused together, like we have right here, into what we refer to as compound pistils. So there's only 1 pistil here, because it's 1 entity, it's 1 thing, 1 structure. But if you cut into that, it's made up of 3 carpels, so it's compound pistil. All that again should be reviewed from lab. And how do you oftentimes figure out how many carpels you have if you don't want to cut the flower? If it's a compound pistil. Exactly right. Count the number of lobes on the stigma, right? That will help you with that. All right, now what's the advantage of having this carpel? Well, one of them is actually going to relate to something we discussed earlier. Because the stigma and the style are the receivers of the pollen. They're responsible for catching the pollen. The plant may actually have a bit of control here in what sperm actually fertilizes the egg. Because just because the pollen has gotten to that point, made it to the stigma, doesn't mean that the sperm has been delivered yet. There's another step there. Right? Once it actually gets to the flower, that pollen grain, as you call, has to form a tube. And that tube has to go all the way down the style, down to the bottom of the ovary, and release the sperm before fertilization can even occur. That's going to take some time, right? And the plant, in some cases, can slow that growth. Right? And if it prevents the pollen tube from growing, fertilization can't happen, the sperm can't be delivered. Or it may slow it down enough that something else, another tube that's going down, may actually be able to beat them there. It's a race to fertilize the egg. Now this, what we talked about before, we have this inhibitory effect, where it's chemicals in the style stigma that's doing this. It could be between the pollen and the stigma of different species, which is what we talked about before with pollen pistil incompatibility. You've got 2 different pollen grains that have landed on the same flower. One's the same species of that flower, the other one's a different. Right? And the style is going to slow the growth of the pollen tube of the one that's a different species. The other one's going to get there first. Okay? That's one way it can happen. But we didn't talk about this. It could also be inhibitory between individuals that are the same species. So one of the things that they're finding is that certain plants will actually prevent pollen tube growth between individuals that are too genetically similar to them. They've got a lot of similarities in their DNA, and there's something about that the chemicals will actually inhibit the growth of the pollen tube from that and prevent the sperm from fertilizing the egg. Why would you want to do that? Well, who's going to have the most similar DNA to you? Your offspring, your siblings, your parents, okay? Your family, right? Okay? This is to prevent inbreeding. Okay? If you can do this, you can prevent inbreeding. And if anybody knows anything about inbreeding is not generally a good thing. Okay? If you keep doing that over and over again, you can run into problems and genetic situations later on. So by doing this between individuals that are the same species, they can actually inhibit inbreeding as well. Okay? So lots of advantages being able to do that. The second, though, is the obvious one. They can make a fruit. They'll form a fruit. And the fruit does many things. One, it protects the seeds because it's inside of that. That's the vessel. Because the carpel now has formed into that fruit. So we got protection from seed. And in some cases, it's also involved in dispersing those seeds to new locations. So the offspring don't have to compete with those parents. How does it do that? Well, it can use animals, it can do water, it can do wind. It really depends. But sometimes parts of the fruits will be modified into structures that help them do this. So we should remember what a fruit is. A fruit is going to be a mature ovary, okay, together along with any floral or vegetative structures that ripen and enlarge with it, okay, as it starts to grow into fruit. So for example, in the apple, we didn't have just the ovary, we also had the stamen, the petals, and the sepals, the hypanthium, that formed into a significant part of the fruit too. So it just depends, right? But that ovary is still going to be involved in it. And it won't form if fertilization doesn't occur. So the plant's not going to have to expend any energy unless it's guaranteed that this could potentially be an offspring. And why put the effort into making something like a fruit like this if the seeds aren't going to get fertilized and could potentially become offspring? And those contain the seeds. So remember, a fruit is different from the seeds. The seeds are inside that. So we got our watermelon, the whole watermelon, that's the fruit, and inside of it, the little black seeds, those are the seeds. Okay, now remember, the ovary wall, and which is part of the fruit, is called pericarp. Now in lab, we actually split it up into different layers too. We had the exocarp, mesocarp, endocarp. I'm not going to do that here for this lecture. We're just going to talk about the ovary wall as a whole and just talk about it as the pericarp. Remember, it can be highly modified for dispersal. You can have hooks and briars or little hooks and sticky substances on the outside of that for dispersal. And if you have a fleshy fruit, which if you recall, pericarp is oftentimes involved in that, the skin layer is usually the exocarp, and the mesocarp is usually the fleshy part, and the endocarp may be either soft or it can be stone-like and droops. Okay? Those are typically animal dispersed. Okay? The seeds may actually get ingested as the whole fruit is eaten and passed through the gut. If it gets through the gut unharmed, which many of them do, because it's actually good for them sometimes to go through the digestive system and have that acid where may that seed go, they can germinate. And if it goes through the gut unharmed, now this organism is going to poop this somewhere else, right? We don't eat and then we wait a couple days and then poop it out in the same spot where you were just sitting in. We wander around and do other stuff, and eventually it's going to get to a new location, so it's going to get dispersed in the process if it passes through the gut in the digestive system. Or it could even get spilled during the eating process. Maybe they don't actually eat the seed, they just eat the fruit part around it, and the seeds get dropped out of it in the process of eating that. Now dry ones can be dispersed by wind, animals, or water. So because this cockle burr right here is still a dry one, and yet we're still using animals to disperse that. So it doesn't have to be just a fleshy fruit for animals to disperse it. You can have dry fruits that are dispersed by animals too. And this one, we'll get into this one later, this is the coconut, right? Coconut's float. Dispersed by water, one of the few ones that still uses that. Okay. Now, I also talked about how for angiosperms, we're gonna have reduced gametophytes on both the male and the female sides. They're gonna be really small, made up of just a few cells. The male one's the smallest of all. Okay? The male gametophyte in angiosperms is gonna start as 2 cells, and eventually it's gonna end up as 3. And we'll walk ourselves through the process. And for these, I do want you to know the names of the cells. Again, I told you for this semester, we're gonna be going through a lot of different cells. There's only 2 that you have to know for the pollen grain. Right? There's going to be only 2 that you have to know for the female side too, even though there's going to be 7 total. All right, so how does this form? Well, we got to backtrack a little bit, go back in our steps of the alternation regeneration life cycle, and we're going to go all the way back to the microspore, because that's ultimately what our pollen grain develops from. So we've got our microspore. It divides mitotically, because it's basically growing. Becoming multicellular, and it forms 2 cells. Here's what they're called. We have the tube cell, aptly named because it's going to be what makes the pollen tube, hence the name tube cell. And we have the generative cell. That one is going to be responsible for making the sperm, kind of an important one. Those 2 cells together, combined, make up the pollen grain. So if you want to talk about them in combination, that's the pollen grain. Here's what it looks like. The tube cell is much bigger. The generative cell is actually inside of it. So there's the generative cell. This whole thing is the tube cell. Now, in the second picture, you can actually see the pollen tube forming from that big tube cell right there. But you'll notice that something's happened to the generative cell too. Okay, this is what it started as. Now we end up with 2 cells. That generative cell is going to divide once, and it's going to make 2 sperm. Both of which are non-motile. These are plants that do not have flagella on their sperm. They do not need water for fertilization to occur. Sperm doesn't have to swim to the egg. It doesn't need to because it's going to get delivered by the pollen grain. Now, when that pollen tube or when this division occurs is going to depend on species. Sometimes it happens after the pollen's released. Sometimes it will happen before.